Hello, City Council. My name is Isaac Darden, and I would like to address the recent discovery that I have made after the events that have taken place on the local beaches in the finding of dead fish. I have discovered that the chemical toxin responsible for this is called antimycin A, also known as Fintrol. So, firstly, what I'd like to get into is the chemical structure and the functional groups associated with antimycin A. So antimycin A contains a large ring system with groups of atoms attached to the ring itself. Antimycin A contains 28 carbon atoms, 40 hydrogen atoms, 2 nitrogen atoms, and 9 oxygen atoms. Antimycin A is a lactone and is associated with the functional groups of formamides, benzamides, and phenols. So secondly, we'll talk about the usage of antimycin A and the effects that it has on organisms. So antimycin A is produced by Streptomyces bacteria and is used as a commercial fish poison. And the following is my hypothesis on how antimycin A affects the cells in organisms. So ATP molecules are synthesized within mitochondria during the aerobic cellular respiration. ETC provides transporters required for ATP synthesis within the inner mitochondrial membrane. ETC then performs the last step of aerobic respiration and divides into complexes 1 through 4. Complex 3 of ETC, known as cytochrome oxidoreductase, supplies electrons to complex 4 by reduction and oxidation of iron molecules. Antimycin A is secreted by Streptomyces as a secondary metabolite. And finally, antimycin A binds itself to cytochrome C reductase enzyme present in complex 3 in ETC and inhibits it, therefore blocking the process of cellular respiration. And so just to kind of summarize the impacts of antimycin A, the cellular respiration process is blocked. Without cellular respiration, oxygen levels deplete in the organisms, and the mitochondria is the organelle affected and targeted in this process of disruption. So next I want to kind of break down how antimycin A affects metabolism. So simply put, antimycin A blocks mitochondrial ATP production by transporting fluids, phosphate, and glucose. Ultimately, this inhibits the mitochondrial oxidative metabolism. In the process of disrupting the mitochondrial respiratory chain, antimycin A binds itself to the quinone reduction site of the cytochrome enzyme. So some of the possible causes antimycin found in the water is antimycin is a restricted pesticide used to kill unwanted fish from ponds and lakes. It is likely that someone illegally attempted to use antimycin to get rid of a specific species of fish for some other reason or for some other reason that is unknown. It is important for the public to know that this chemical degrades in 2 to 7 days after use, therein which it will no longer pose a threat to the health of the water sources of fish and human activity. And on that note, it is important that the public knows that antimycin A is harmful to humans and can irritate eyes, skin, and can cause respiratory issues. So it is very important to let this chemical degrade in the 2 to 7 days before going back into the water and going about your regular water activities. That would conclude my findings on the chemical that we found. Thank you very much for your time, City Council. Bye-bye.